materials in, specific ligands into the cell, and that can be done through receptor-mediated endocytosis. Okay? Just like we can bring materials into the cell, sometimes we need to remove things from the cell. Okay? So when we're kicking things out of the cell, it's called exocytosis. Okay? So it's essentially the opposite of endocytosis, but it is a way of exporting material out of the cell. This could be material that the cell made, it could be waste material, it all kind of depends. Okay, let's talk about the organelles more specifically. So organelles you can think of organs. We all know what organs are. We've got hearts and lungs and kidneys and those sort of things. Different parts of the body that function to make the organism work as a whole. At the cellular level, we have organelles, little organs, that perform functions to grow, maintain, and reproduce the cell. You guys covered a lot of this in the lab in week 3, so hopefully this is all a review. Okay. Here is a generalized photo of a cell. So on the outside we've got the plasma membrane. On the inside we have a giant enclosed ball that we call the nucleus. It's enclosed by the nuclear envelope. Within the nucleus we have the chromatin, which is the genetic material, and the nucleolus. Surrounding the nucleus, we have the rough endoplasmic reticulum, or called the rough ER. The rough ER is speckled in ribosomes. Ribosomes are what make our proteins. Okay. On the edges of our rough ER, we have the smooth endoplasmic reticulum, the smooth ER. We also have the Golgi apparatus, which is going to be responsible for packaging. We also have things like the mitochondria, which is our energy. And we've got lots of cytoskeletal elements, okay? And we're going to go through all of these individually. Okay, the nucleus. Okay? The nucleus is one of the larger organelles in the cell. And what's really important about the nucleus is it contains the genetic material, or the genes, for each of the cell. Every gene, or every cell in the body, with the exception of the red blood cells, which have it at one point, they just kick it out, has genetic material. Okay, so the genetic material is the blueprint of the cell, and that's going to tell the cell how to operate, when, where, and why. Okay, it is surrounded by a nuclear envelope, so it's got its own plasma membrane that is full of nuclear pores that allow material to come in and out, but it's going to always keep the genetic material inside the cell, inside the nucleus, and it's going to pass messenger RNA on the outside in order to do its job. At the center of the nucleus, it's contained within the nucleolus, okay? And we'll talk about what the nucleolus does here in a little bit. Okay, this is a close-up image of the nuclear envelope. Okay, so like I said, it's a double-layer membrane, very similar to the plasma membrane, but it contains lots of these pores. These nuclear pores are going to allow specific substances to go in and out. We want to keep that genetic material safe and allow things to go out. Okay. Inside the nucleus is the nucleolus. The nucleolus is to produce ribosomes. Ribosomes, I mentioned, are studded throughout the rough ER. Okay, ribosomes are found throughout the cytoplasm as well, but most of them are on the rough ER. The job of a ribosome is to make proteins. Proteins are very important for cellular and body development processes, and so we create those ribosomes inside the nucleus, and then once the ribosomes are formed, they're going to leave through the nuclear pores and float around and either connect to the rough ER or float around the cytoplasm. So the rough ER is a network of folded membranes and its main job is for protein synthesis. Okay, so it's going to take all of that information from the nucleus, and it's going to report it to the ribosomes, and those ribosomes are going to take that information and create the proteins. Okay, it's kind of just a temporary holding facility for all of these molecules, protects them while they're being formed. Outside of that, we have another tubulous membrane called the smooth ER. The difference between the rough ER and the smooth ER are the ribosomes. So the smooth ER does not have any ribosomes. And so that is where fatty acids, phospholipids, and steroids are produced. Okay, so the rough ER has ribosomes, it makes proteins. The smooth ER does not have ribosomes, so it makes fatty acids, phospholipids, and steroids. The Golgi complex, or the Golgi apparatus, it's the same thing, we just like to call it different names to be fun, okay? It is the shipping and packaging part of the cell. It is what's going to package and deliver these proteins and lipids to the plasma membrane in order to excrete them into the extracellular matrix. So it's going to form lysosomes and vesicles, which are little carrying balls of plasma membrane that are going to float around and go through exocytosis in order to release their contents into the environment. So here's an example. Okay, so the rough ER is going to utilize its ribosomes in order to make a bunch of proteins. Those proteins are going to float into the Golgi apparatus. That Golgi apparatus is going to package them into lysosomes or vesicles, and then those packaged vesicles are going to float through the float through the cytosol to the plasma membrane and go through exocytosis to secrete those proteins into the environment. Okay, I mentioned lysosomes. Lysosomes are membranes that are enclosed vesicles. Okay, lysosomes contain digestive enzymes. Those enzymes are there in order to digest and break down things like bacteria, old organelles, those sort of substances. Okay, so we need a way to get these large particles, macromolecules, and fluids across the plasma membrane. We can do that through vesicular transport. This is going to require energy in the form of ATP. Okay, this vesicular transport, or what we've been talking about, the exocytosis, which is moving out of the cells, endocytosis, moving into the cells, transcytosis, which is transporting in, across, and then out. And then we can do vesicular trafficking, which is transporting cellular materials from the organelle in the cell to another. Okay, peroxisomes. Peroxisomes are membrane-bound organelles that contain substances of enzymes that are there to detoxify harmful substances and lipid metabolism. Okay, so peroxisomes are going to break down bad things. They're very similar to vesicles. They're just a little bit different. We have talked about endocytosis, and so we're going to add endocytosis to transcytosis, and so we're going to bring in a material through endocytosis. We're going to do whatever metabolism or change that we need to do. Once it's completed, we can break it off and we can package whatever we need to leave in with the lysosome, and then we can exocytose it. We talked about endocytosis already. Phagocytosis is the act of engulfing solids. And I mentioned that macrophages and white blood cells do this. Just another review on endocytosis. Pinocytosis is when we're sucking in fluids, so when we use water. And I talked about cell-mediated endocytosis, which is going to bring in labeled materials. Okay, here's just another review on exocytosis. So this is how we get hormones secreted out of our cells. We don't want that hormone floating around the cytoplasm, so when the hormone's created, it's going to be packaged and released through exocytosis. Just another graphic there. Okay, let's move on to the next organelle, the mitochondria. Okay, the mitochondria is a double membrane organelle. Okay, so it's got two membranes instead of just the single membrane that we've been talking about. Okay, the outer mitochondrial membrane is an encasement, and within that, it has a highly folded inside membrane that we call cristae. Okay, we're going to learn eventually that it utilizes this inside membrane to maintain lots of different concentration gradients. It is what is responsible for making our cellular energy in the form of ATP. So it goes through this act called cellular respiration, and it's utilizing these different membranes to make energy through concentration gradients. Okay, so we've got the mitochondria, the inside mitochondrial membrane. The folds are called cristae. Mitochondria contains its own DNA that is separate from the cellular DNA. It's got matrix and ribosomes. Okay, another part of the cytoplasm are cytoskeletons. Cytoskeletons are filamental or filamentous proteins that allow cellular movements and support. So it's going to allow these packaged materials or materials in general to move throughout the cell, and it's going to maintain the shape of the cell, whether it needs to be a circle or a square, those sort of things. Okay, we have three different types of the cytoskeletal filaments. The first one we're going to talk about is microfilaments. Microfilaments are rod-like structures. They are made of actin. Okay, their job is to help the cells change shape. Sometimes, like white blood cells, they need to roll around or ooze across a substance. And it's these microfilaments that are going to allow the entire cell to change shape. We have intermediate filaments. These are rope-like structures that are made from fibrous proteins. Okay, the function of these are reinforcing rods, so they're going to anchor the cell to maintain shape and to keep the organelles in their specific places. The third type are microtubules. These are straight hollow tubes that make up the rigidity and locomotion appendages. Okay, so not all cells, but some cells have different appendages or things that stick outside of them that allow movement to happen. We call these flagella or cilia. So a sperm, for example, has a flagella. It's a tail. And some cells in our body have cilia, which are little hair-like appendages that are going to allow movement to happen throughout the body. These are all made up of microtubules. Okay, I'm going to stop you guys there as we leave talking about the cell and going on to more cellular components. So I'm going to pick up with the rest of it in the next video. Thanks, guys.